Well, hello and welcome to Bio 212, your Anatomy and Physiology 2 at Norwalk Community College. And we are beginning Chapter 19, the cardiovascular system. And within the cardiovascular system, particularly inside Chapter 19, what is blood? The connective tissues that are liquid. Remember, connective tissue arises from mesoderm, and that means what we're actually talking about is what actually is the purpose of blood. And there are three general functions, right? So you can see here the three general functions: transportation, regulation, and protection. Okay, so transportation. We're thinking about what are we actually transporting here. Everybody knows we're going to be transporting oxygen, right? So O2, yay, no problem with that. Okay, we're also going to be transporting carbon dioxide. Okay, we'll learn about that in a lot more detail, probably more than you want to know. And we also transport nitric oxide. These are all expected and normal materials to bind to the protein that we know as hemoglobin. It's actually a complex of 4 proteins that are bound together, and they carry each of these in different amounts and different states depending on what's going on in the body. Okay, what you don't want to carry on this, of course, is carbon monoxide, and we'll come back to that later when we talk about the structure of hemoglobin. Nutrients. For just, AMP1 level here. C6, H12, O6. You know that is glucose. We know we can change the concentration of that glucose depending on the hormones inside the bloodstream. Look at that right there. Okay, and that's important because you're also going to be having the breakdown products of that. And guess what? One of the breakdown products of that is, of course, is this over here, right? And you can see how it all begins to tie together. Let's think about those hormones for just a second. Every single one of the hormones that is either water soluble or non-water soluble, meaning lipid soluble, that's inside now the back of your mind, that everywhere inside your circulatory system, you could be transporting a hormone. We also use blood for the regulation of pH inside of the body. You have a normal range of pH inside your body, 7.45 to 4, sorry about that, to 7.35. And we spend a whole lot of time talking about how that happens, not necessarily inside this chapter, but as the rest of the semester goes along. And you have a temperature range where that temperature range can't be ignored, because below specific temperatures, all of the metabolic processes inside of your body tend to cease. And if they get to be too warm, the efficiency and the ability to regulate the complexity, of course, is lost. In behind all of this, though, is we regulate osmotic pressure. So osmotic pressure, of course, is the movement of water into a space as a result of the change of the number of solutes inside of that space. And what did we have up here? Well, we have nutrients. And you're going to see down here, we actually have proteins as well as cells. And that is what actually determines osmotic pressure inside of our circulatory system. Finally, we have this idea of protection, where clotting is a series of mechanisms we get to at the end of the chapter, towards the end of the chapter, where we actually take insoluble proteins, insoluble, just put in or not soluble, so NS, and then we make them soluble, or vice versa. Okay, and you'll see primarily it's the actual opposite of this. We take soluble proteins such as, say, fibrinogen, and we make them insoluble. Okay, you can also make soluble proteins become insoluble. You'll see how that happens later. We have a family of white blood cells ranging from neutrophils to basophils. They each have a specific responsibility, but one thing that they are definitely not doing is they are not carrying oxygen. And then finally, we have a whole series of proteins. We're going to come to a description of, or at least a list of them in a couple of minutes. But again, when you're looking at the proteins, they all have their own biological responsibility, bio, right? But the consequence of them simply being inside your circulatory system means that they're going to regulate osmotic pressure inside the body. So one way to think about this is if there's no proteins inside your bloodstream, what happens to the osmotic pressure? If you have too many proteins inside your bloodstream, what is the consequence? Likewise, if you have too few red blood cells or too few white blood cells, you can actually see what's going to happen. Okay? So that's an introduction. You know, these are the things we're going to be thinking about as we proceed through this chapter. Let's get rid of that. Let's go over here. Blood plasma itself, blood plasma, is essentially a liquid matrix that is extracellular, meaning outside of the cells, and it's essentially water. It's 91.5% water. So if we took some volume of that out of you, you'd see a few things that would be floating around in it, and that's about 8.5% of that is solute. The liver cells, hepatocyte liver cells, okay, synthesize most of the plasma proteins, the albumins, the fibrinogens, the antibodies. And here I put in the word consequence. And what I want you to start actively thinking about is if we change the concentration of any of these, meaning you increase 8.5% or decrease 8.5%, what is the consequence? And as I mentioned earlier, you're going to have other materials inside of there. Primarily what we spoke about were the hormones, gases, and waste products, right? But there's also that glucose a moment ago we spoke about, but that could also be fructose. Remember, it could be lactose. It could be all sorts of stuff you've ingested, but will be converted into something else inside your liver. You can see there's a whole host of enzymes that are floating around inside the liquid extracellular matrix. And the thing that I need you to start entertaining the most is what is the concentration of electrolytes inside of that 91.5% of the water inside of your body. Okay? When we're actually thinking about blood, we're thinking about what's inside of it, those formed elements, the cells and the fragments. Red blood cells obviously make plenty of sense. White blood cells, you might know most of them already in terms of vocabulary from A&P1, but the actual responsibilities we delve into a little bit deeper in this chapter. We definitely introduce them. They're developed in their own chapter, chapter 23, I believe. Okay, no, 22. And then they're platelets. Platelets are, of course, fragments of a specific type of progenitor white blood cell, and they are going to help us regulate clotting inside the body. Let's take a closer look at what's actually inside of the plasma of your blood. Okay, meaning if we take out blood, we can break it up into plasma, red blood cells, and the buffy coat. And if you do the math here, you'll see it gets somewhere close enough for us to pay attention to. But what I'm hoping people are noticing when you look at this more actively is that if this is you as a human being, let's put you in here, right? This is you. And you may not want to think about this like this, but I don't really care. There we go. Put an eyeball there, put an eyeball there, put a smiley face there. And somebody put a curly Q for hair on their practical or their quiz or something like that. I thought that was pretty awesome. Okay, and notice what we're actually describing here. Your mass, your body weight, okay? And your body weight is essentially whole blood, which is about 8% of you, and all of the other fluids and tissues inside of your body. This is the mass. This is when we're trying to figure out what dosage of drug to give you, okay, what we're thinking about is what does that drug need to be dissolved into? Okay? Does that make sense? I hope so. Because if we pull out some of that blood, you're going to see that, yeah, you have red blood cells. They take up a specific mass. You have these white blood cells and platelets over here. But this really is the volume of materials we can deliver drugs into. And it's not necessarily the only way that it works, but it's definitely one of the more important ways to think about it. That blood plasma, that 55%, is broken up into 7% proteins, 91.5% water, and 1.5% of those other solutes. Those proteins are the albumins. These are made inside the liver, and they are primarily used for regulating solubility and osmolarity inside of the body. Globulins will have different responsibilities, but immunoglobulin might mean something to you where you have soluble proteins. You might know them as antibodies or members of the family. And they, if you change their concentration, will change the solubility of the, I should say, the osmolarity of the plasma of blood. Fibrinogen is the precursor protein for clotting mechanisms that we get into at the end of this chapter. Okay, and there are others, but this is a pretty good representation of what you actually find as proteins. Notice there, definitely no enzymes right here, but they're going to be something inside there at some point in time. There's the water, and then we have the 1.5. And what I want you guys to be thinking about more and more as the semester goes along are what are the electrolytes we actually find inside of this bag of salt water. Nutrients, again, glucose, fructose, whatever. Gases, carbon dioxide, CO2, as well as O2, and to a much lesser extent, NO. Okay. Regulatory substances, okay, we'll just say those are hormones. And then the waste products, again, that's CO2 as well. All right, so you can see what's actually happening here. We're beginning to break down, hopefully, what's actually inside of the plasma portion of all of this. The formed elements, they get their own treatment. That's 45%. This is a mixture of red and white blood cells, anywhere between 150,000 to 400,000 platelets per, look down here, look at the units, microliter. That's 10 to the minus 6 liters. 10 to the minus 6 liters. So you've got anywhere between 150 and 400,000 platelets per microliter. You have white blood cells between 5,000 and 10,000 white blood cells. You'll see you actually have this different family of them. We'll come to the families in a couple minutes. But here's where you need to start paying really close attention. On average, you have between 4.8 and 5.4 million red blood cells per microliter of blood. Okay? You'll see why that's really important when we start thinking about how much oxygen we're actually moving around. Okay? Cool. Just take a quick look at the sort of the representation of white blood cells. Most of the white blood cells inside of your blood are neutrophils. Then you have these things that are called lymphocytes. Then you have monocytes. Then you have eosinophils, and then you have basophils. Okay? It is important that you understand this ratio because anytime this ratio changes, you can use it as a diagnostic tool. Okay? If this number falls, it means something. If this number raises, it means something. Likewise, you can go through each one of these and figure out what your body's actually doing as you increase or decrease these numbers. Okay? So let's go like that, and let's come down here, and let's take a closer look at what this looks like. You're going to see this inside lab. You will not ever see anything, anything so precise as 30,500 times magnified, because this is a scanning electron micrograph of a small piece of blood where you have white blood cells over here, platelet over there, and these are red blood cells. Instead, what you're going to see is something like this, where, again, at 400 times total low magnification, you can see the plasma, which is the space. Notice you can't tell that it's probably straw yellow colored, but it's space. You have red blood cells, and you can see this biconcave disc. You know it's biconcave because it's more hollow in the middle. Take a look at the indentation here. You'll see why that happens in a few minutes. Here's a platelet over here. Here's different types of white blood cells. And notice, even at the beginning, if I told you just look at this quickly and then look away, you're going to see the white blood cells will differentiate themselves as a result of how the nucleus, one, is formed, and two, can you see granules inside of these structures or not? And in this case, you should be able to see granules inside the neutrophil. Okay. There's one more thing before we move on. Bring out the pen. You have a ruler here, and the ruler is, and I don't mean like ruler of the world. I mean, you have a distance that you can use. Primarily, most red blood cells are about 8 microns across diameter, right? So that means, even though you'd have somebody inside the room who will argue this with me, well, you know, they're not all 8 microns, so how am I supposed to use it? Generally speaking, the average is about 8 microns in size. So that means now you can figure out the size of, say, a platelet, or you can figure out the size of a white blood cell. These are tools you want to acquire as you're going along, as you work your way through this. Okay? Let's get rid of that. Formation of blood cells is by negative feedback systems. So the number gets to be too low or the number gets to be too high, and some signal will actually increase or decrease the number of red blood cells and platelets inside circulation. Likewise, the abundance of white blood cells is based on the response of the pathogen or the foreign antigens. Okay, like it's its own chapter, so don't freak out about that yet. But hemopoiesis or hematopoiesis occurs inside the red bone marrow, inside that non-compact bone or trabecular bone, and arises from pluripotent stem cells. So meaning all of these are going to arise from pluripotent stem cells. Whoa, what happened there? Control-C. And they're going to differentiate into all of these different stem cell types. Okay, so again, it's connective tissue, so therefore the mesoderm gives rise to mesenchyme. Mesenchyme is going to be these pluripotent stem cells. And then from here, what we can do is we can go over there and take a look at the cartoon to make sure you understand this. Here's that pluripotent stem cell. We know we're inside the bone marrow, and immediately you can see two arrows here. These two arrows suggest that by definition, the default pathway is a lymphoid stem cell. And that's because there are no multiple layers of regulation telling myeloid stem cells what to become next. So pluripotent stem cells, if left to their own devices, become lymphoid stem cells. And you're going to notice that they're all blast type cells, okay, which means that they are all immature. You have a T lymphoblast, a B lymphoblast, and an NK lymphoblast. Okay, so these are T cells, B cells, and natural killer cells. You'll see what they are down here if you follow along. This is the formed element that's actually inside the bloodstream. T cell, B cell, or natural killer cell. But because they are blast cells, it means they have to mature. They all have their own mechanism for doing that. That's outside of what we're talking about inside chapter 19. But you can see that natural killer cells are going to be doing one thing, B cells do another thing, and T cells will do another responsibility. What I want you to pay attention to is further differentiation creates a unique tissue here, and that unique tissue is this thing that's called a plasma cell, and these plasma cells make antibodies, and these antibodies were those globulins that you saw inside the bloodstream a few slides ago, right? And this is significant because it means now we can actually increase the number of antibody-producing cells inside of your body dependent upon the pathogen that it identified. Okay. If we now look over here to the myeloid stem cell line, you can see that in the presence of specific hormones, meaning inside the bone, meaning now bone is an endocrine gland, you have colony-forming units for erythrocytes, megakaryocytes, and granulocytes. And what this means is CFUE will give us erythrocyte differentiation and development, or development and differentiation. Megakaryocytes, say megakaryocyte colony-forming unit, will give us a megakaryoblast, which gives us a megakaryocyte, and it's this megakaryocyte that splinters up into platelets, which will then be called thrombocytes. That's the little platelets you saw inside the blood a moment ago. The myeloid stem cells, when you're looking over here, we have eosinophilic myoblasts, basophilic myoblasts, okay, that are the actual default pathway here, where you have either an eosinophil and a basophil. And you can see that these actually have little granules inside of them, just like basophils do here, and the neutrophil does over here. Okay, so we would collectively call these granular leukocytes. Okay, so let me put a line underneath that. And you're going to see that even though GFU can give rise to myeloblasts and monoblasts, the neutrophil and the monocyte are different in that the monocyte is an agranular leukocyte, along with the T lymphocyte, the B lymphocyte, and the natural killer cell. And similar to our plasma cell over here, our monocytes, when activated by the presence of a specific pathogen, okay, will then differentiate into this cell that's called the macrophage, and it actually is what runs around the body in sort of doing its job of clearing whatever those pathogens are. Okay, so this is a pretty good place to pause, and we'll pick up in the next set of slides. We're only on slide number 6, looking at sort of how did this happen and what we're going to do with it next. Okay.